Hello, and welcome to an easy overview of prenatal brain development and what the basis for our brain to reach its full potential looks like. Our first part is centered on the neurulation, or the formation of the neural tube. 16 days after fertilization, three layers begin to develop called the endoderm, the mesoderm, and the ectoderm. After 2 weeks, the ectoderm forms the neural plate, which is the starting point for the nervous system. As the plate folds into itself, it forms a neural groove and a neural folds. By the third week, these folds fuse, becoming the neural tube, which then becomes the central nervous system. Cells from the neural folds then separate from the ectoderm and forms a cluster of cells. The cluster of cells are known as the neural crest. From the neural crest, cells migrate to form parts of the peripheral nervous system, or the PNS. The second part that we will be looking at is differentiation and brain vesicles. The neural tube differentiates into three primary vesicles: prosencephalon, the mesencephalon, and the rhombencephalon. These three vesicles are also known as the forebrain, the midbrain, and the hindbrain. These primary vesicles continue to develop into five secondary vesicles. The prosencephalon forms into the telencephalon and the diencephalon. The mesencephalon stays as the mesencephalon, and the rhombencephalon becomes the metencephalon and the myelencephalon. The telencephalon becomes the cerebrum. The diencephalon forms the thalamus and the hypothalamus. The mesencephalon, or the midbrain, stays the same. The metencephalon forms the cerebellum and the brainstem. The myelencephalon becomes the medulla oblongata. The next step that we will be looking at is neuronal proliferation and migration. Deep inside our brain, as it develops, there are special cells called progenitor cells, which act like builders and are the foundation for most things in the brain as it develops. They live in two key areas: the ventricular zone, VZ, and the subventricular zone, SVZ, where they rapidly divide and generate billions of neurons. Once these neurons are formed, they move into different places where they belong, sort of like fitting a puzzle piece into a larger picture. These neurons end up moving in two distinct ways: radial migration and tangential migration. Radial migration goes vertical, using radial glial cells as guides to move up and down, while tangential migration is like moving sideways. Neurons travel horizontally across different areas of the brain to form structures like the basal ganglia. These migration methods ensure neurons get to exactly where they need to be to help shape the brain's networks and functions. As the brain completes its development, it organizes into several major divisions that each play a different crucial role: the cerebrum, the thalamus, the hypothalamus, the brainstem, and the cerebellum. The journey our brain goes from being a single cell to a fully formed brain that allows us to operate our everyday lives. It's incredible. Even after the incredible growth that rapidly happens in our brain, its final stages of growth last well into our 20s. Our brain is a masterpiece of nature that constantly learns, adapts, and shapes who we are. It all starts with one cell. Thank you for watching. I hope you enjoyed learning about the incredible journey of brain development. Until then, see you next time.